I think Joe Bertino asked the question that led into the next talk, indeed, by Curt Civin. Curt Civin is a professor of pediatric oncology at Johns Hopkins School of Medicine. He has for many years been a prime mover in the elucidation of our understanding of hematopoiesis and differentiation in the hematopoietic pathway. Indeed was one of the pioneers in identifying CD34 as a major cell surface marker for the hematopoietic stem cell. And we are reminded with the previous work, the 21 years of Ihor's work and Curt's work, that stem cell biology really begins with hematopoiesis. And the best example, or one of the best examples of stem cells, came from Till and McCullough and their pioneering experiments that said there was indeed a hematopoietic stem cell and that the regeneration of 15 or 20 different cell and tissue types came from hematopoiesis. And Curt has been one of the leaders in this area, and he will address the topic of microRNAs, the Maestros of hematopoiesis. Curt, it's a real pleasure to have you here.
Thank you, Arnie. I want to add my thanks to the organizers and the elucidators and controllers of the weather for this beautiful day, Shay Einstein. And it's just very important to be in New Jersey, a state that vigorously supports both STEM and cancer cell research, and I know that's very important to all the speakers and to this audience. It's also a pleasure and a challenge to follow my friend Ihor Lemishka. I think Ihor and I met about 20 years ago at a Gordon conference, and there we walked around scenic New Hampshire on days like this, and we were both fascinated with the practical ability to study primary cells from a developing, and that was the hematopoietic system, in mouse and in humans. And since then, I've had a couple nice opportunities to write letters for Ihor, very easy letters to write, because he always elucidates with novel technologies things that we've been wondering about for years, and then we go back and teach them to our students. And then he really sets up the questions that we're going to investigate next. So if I can advance the slides with the green button, I can thank Ihor and also then tell you, as my university requires, that I have a self-interest in stem cell biology via the CD34 molecule. So anything I say about that, or maybe about stem cells, you should take with an extra grain of salt. And what I'll talk about today is largely studies of the hematopoietic system, which, as you all know, I think, is a wonderful paradigm for thinking about stem cell biology today. And yet, even today, after 50 and more years since Till and McCullough, really little is known about the exact molecular mechanisms. Certainly the systems biology of hematopoiesis is totally unknown. In general, we can knock out a lot of genes and get no hematopoiesis, but we still have no great idea of how to make the hematopoietic equivalent. Still today, we have no great idea of how to make the hematopoietic equivalent of an iPS cell that could, outside of the body, generate lots of hematopoietic stem cells from one. In vitro, when we take a primary hematopoietic stem cell, as many of you know, and we give it all the goodies that we can find from the genome, still these cells proliferate only at the price of differentiation. We can get them to do a couple of self-renewing divisions or semi-self-renewing divisions with the growth factors that I have in blue up there, stem cell factor, thrombopoietin, FLT3 ligand, and we can get genes into them, but we really can't get them to grow us up a pot of stem cells that we could further study in vitro, and actually this is a characteristic of all so-called adult stem cells that we still have to elucidate. So several years ago, again, following I think in Ihor's and some others' footsteps, we said that we had to get back to basics and really take isolated human hematopoietic stem cell populations and study them transcriptomically, and we'd figure out we'd identify the important molecules, study them, and be able to then further elucidate the basic stem cell processes of course, self-renewal and differentiation, but also quiescence and homing in addition to fate specification. And of course, the translational goal here is to make high-performance stem cells and grow them up in a vat so that we could transplant not one patient, but hundreds of patients from one bone marrow donation. Can't do that yet. So Bob Georgantis here, looking intimidated by the project, took huge amounts of CD34 cells from human cord blood, human bone marrow, and mobilized, so-called mobilized blood that's used for transplantation in patients, and purified a stem cell enriched versus a progenitor enriched population. And you see those pictures up here. So these are not pure stem cells and not pure progenitor cells. They're enriched in the respective populations. And then we did Affy arrays, as Ihor described, and looked at the genes expressed. And Bob did a very smart thing by studying 3 sources of stem cells. He was able to make a Venn diagram like this, and to cherry pick, looked at the genes that were overexpressed in the stem versus the progenitor cells of all 3 populations. And there are about 80 of these genes that were overexpressed in all 3 tissues. We really still haven't concentrated on looking at the 90 or so genes that were downregulated in stem cells versus progenitor cells. And we confirmed this globally by the SAGE technique and individually by about 150 PCRs for several of the important genes. And then we started to look at the list, do sort of a meta-analysis with Ihor's paper and another paper from the Mulligan lab and Melton lab, and looked at some of these 81 genes and who were they, and were they found on other people's arrays of human and mouse stem versus progenitor cells, and narrowed down on about 15 interesting genes, many of them transcription factors, but some of them also, and I'll talk about this in a few minutes, a potential stem cell markers in that they were surface membrane proteins. One of the genes is a gene that Ihor mentioned, KLF4 here in the human, and this was a gene that I'm going to tell you a little story about, drilling down on its function. This is the work of John Alder for his graduate thesis in my lab. And John looked at those genes and said, well, this KLF family is interesting. The parent gene is in flies, and it's interesting in flies. Humans have about 25 KLF family genes, and the KLF4 gene is a member of a clade whose very, very close relatives are KLF1 and 2, and there are structural features in common for all the KLFs and also for this clade that are very closely similar. Well, KLF4 is known to be important was known to be important in erythroid development, KLF2 in T-cell development, KLF3 in myeloid development, KLF6 recently in stem cells, and since we found KLF4 overexpressed, this is PCR confirming, in human and mouse stem cells isolated versus progenitor cells, we thought it might be important up here in stem cells. By the way, on arrays and confirming techniques, KLF4 is also highly expressed in mesenchymal stem cells from human, in H1 and H9, 2 lineages of human embryonic stem cells, and in many samples of leukemia stem cells. So something worth studying. So John made some tools. The first tool he made was an overexpression tool And so he made in some contexts, a regulatable KLF4 driven by the F1 promoter with a marker gene, GFP, driven in this slide by the CMV promoter, but using other better promoters in some other experiments, and showed that the expression of GFP using a lentivirus to introduce this gene in these markers, that the introduced amount of KLF4 was directly proportional to the GFP signal on a protein level. And then he took KLF4 and he forced its expression. So he said, okay, KLF4 is meant to be high in stem cells and then to go down in progenitor cells. What if we keep it up in the progenitor cells by just artificially overexpressing it? And when John did that, he found that he knocked out basically the ability of cells to form hematopoietic colonies from mouse or from human cells. Colonies could not be generated or were generated at a few percent that corresponded to very, very low levels of transduced KLF4. He looked in stem cell assays as well, which is a neat feature of hematopoiesis. You can do in vivo assays, both in mouse and in humans. So you can, in mouse, take mouse cells and transduce them with KLF4 and with a regulatable promoter, even use tamoxifen to induce expression after transplant. And you can do this in either a congenic mouse, or you can also do this in an immunodeficient mouse that I'll tell you a little later, and use human stem cells, stem progenitor cells, to transduce and transplant. And what he found in both cases was that the contribution of the KLF-containing cells quickly went to 0. So this KLF4 overexpression drove the cells, when expressed in progenitors, you got no hematopoietic output. And consistent with this, we couldn't generate stable cell lines expressing KLF4, had to use the estrogen receptor regulatable construct to get that in cells, and showed that basically various cell lines were also, their growth was inhibited by expression of KLF4. We looked at the mechanism of this. It looked to be a G1 arrest, and the important molecule looks to be P21, as has been shown in several other systems that KLF4 regulates P21. In these cells, it does as well. So enforced KLF4 inhibited hematopoietic proliferation. What about knockout? So KLF4 knockout mice we got from Julie Segre at NIH. And the trouble with these mice, from the point of view of studying their adult biology of KLF4, is that the mice die at birth from skin problems, dehydration, and infection. On the other hand, an advantage for the hematopoietic system is you can take their fetal livers, and these fetal livers contained hematopoietic stem cells, and so you can study hematopoiesis in the fetal liver. First, John looked at the total numbers of cells in the fetal liver. They were the same in the wild type and the knockout mouse, and the het was the same. And then looked at colony formation from the fetal livers, and they were the same in wild type and knockout, so disappointing. But he persevered, and he transplanted those cells into a congenic mouse that was differentially marked natively, and so he could distinguish the cells that bore one isotype of CD45 versus the other strain that bore, the congenic strain that bore CD45.2 isotype, and look at host versus donor. This is just an idealized picture, because what he found was that the wild type were no different than from the knockout mice. So he went on, and he said, well, that's short-term hematopoiesis. What I'm going to find, he thinks, was a defect in long-term hematopoiesis. So he used competitive repopulation assays and followed the mice for about a year. And depressingly, there was no difference between knockout and wild-type mice. So despite the fact that KLF4 was highly expressed in early stem progenitors, and that forced expression inhibited proliferation, likely via P21, KLF4 wasn't essential, and so the next experiments are to breed these mice with knockouts of the close family members KLF2 and KLF1 and see if compensations explains this. This will be complicated because those molecules have hematopoietic effects of their own. And John decided to graduate from the lab and move on to Carol Greider's lab instead of doing that. But before that, he had to get his thesis. And so he said, well, this is disappointing. What else does KLF4 regulate? And he found various monocyte genes. And then he found some papers relating KLF4 to monocytes. And so he said, okay, let me look at KLF4 expression in monocytes. He did KLF1 and KLF2 as a control for his quantitative PCRs, and found that KLF4 was actually highly expressed in monocytes and less highly expressed in NK cells, and that when you induced monocytes with LPS, KLF4 expression went up. So he said maybe there's a link to monocytopoiesis. And without telling you a long story here, I'll just show you these pictures that show that he found the phenotype of the knockout mice, and that KLF4 has the unique phenotype of knocking out KLF4 has the unique phenotype of knocking out monocytes, and particularly a so-called inflammatory monocyte population. And so what he showed was that KL4 is necessary for this subset of inflammatory monocytes that's important in the inflammatory response that we were talking about earlier in various autoimmune diseases, that KL4 is also, he showed in experiments which I didn't show you, is sufficient to induce monocytic differentiation of each HL60 cells. And so we have the idea that since KL4 fuels inflammatory monocytopoiesis, it's a potential target for immune modulation here in the mouse. But if you go back to the big picture, the problem is, okay, that's one of those 81 genes, or maybe one of those 15 genes that we narrowed it down to. So this drilling down process, cherry picking and drilling down, is going to take a long time. In fact, while we've cemented that KLF4 acts here in hematopoiesis, we still don't know what the heck KLF4 is doing overexpressed in the stem cells, and this was where we really began. And Ihor referred to the work of Yamanaka and Thompson and Yanish and others who've made these neat induced pluripotent stem-like cells from fibroblasts. And one of the key 3 or 4 genes that you can use in cocktails to reprogram these cells to embryonic stem-like cells is none other than KLF4. What's it doing there? Certainly not inducing P21 as the key player. And so we don't know the answer to this, but perhaps Ihor's studies, as you heard earlier, are giving us some clues both as to what turns on KLF4 and what KLF4 is doing in all these types of stem cells where we find it. So overall, the gene expression studies have provided us some direction, but again, as Ihor intimated, there are some problems with the transcriptomic analysis. The way we looked at it was that when we looked at a lot of the genes that we know shouldn't be, if we were designing the system, expressed until the progenitor cell stage, we find them at very high levels in the stem cell fractions, as well as the progenitor cell fractions of mouse and of humans. And we could say, why doesn't this tend to cause the cells to differentiate? Why is nature doing this, assuming that the proteins are made? Well, this got us to another piece of data. When we started to look for markers, we looked at some of those stem cell overexpressed genes that were membrane proteins, and some of them had monoclonal antibodies available. So when we stained stem cells with those monoclonal antibodies for which there was huge amounts of message expressed in stem cells and differentially expressed in the stem cells, there was no protein. So this brought us to microRNAs, or microRNAs, the answer to this, to both these conundrums. And you all know about microRNAs. I don't have to talk about this now. And their basic production mechanisms are illustrated here. And it's thought that microRNAs are predominantly translational inhibitors, but they may also act as siRNAs, depending perhaps on the degree of complementarity. This is an evolving area, and microRNAs are new. We're discovering and learning the literature new things every day, so I won't be dogmatic about how they work. What we did, though, was take the current microRNA chips, and Carlo Croce, our friend, had the best chips at the time, we thought, and we collaborated with Carlo and George Kalin to look at the microRNAs expressed in CD34 cells from bone marrow or mobilized blood. We didn't bother with cord blood at this point. And we focused on 32 that were expressed in both these tissues and called these hematopoietic expressed microRNAs, or hemirs. And we looked then at their thermodynamically predicted targets and found that they, at least thermodynamically, might regulate over 15% of the genes that we knew to be expressed by our transcriptomic analysis in stem cell enriched populations. And so, which were real targets. These were just thermodynamic predictions. So we had to do luciferase assays to do this, and basically what you do is take luciferase and put the 3' UTR in whole or in part of your potential target predicted molecule onto luciferase, and then put it into a cell that has the microRNA or that doesn't, or one in which the microRNA has been inhibited by an anti-mir and basically an antisense molecule, and look for an effect of a specific effect of the microRNA. And basically we identified a number of these targets by using the algorithm that we would look only at stem cell expressed genes that were expressed at high levels at the transcriptome level, those mRNAs, and then we would look at the predicted microRNAs, which might be holding those messages in check from making protein, the phenomenon that we were seeing and the model that we were interested in exploiting. And what we're thinking here from these targets, if you look at these cells, you're talking about here verified 18 or 20 verified targets for a given microRNA, at least in these that are relevant in these cells. You're talking about something that can regulate potentially these systems. So in lay terms, perhaps you're talking about, instead of turning on the lights of a set of rooms one at a time, of really flipping the circuit breaker to turn on the whole light, or whole buildings, or in the actual system, whole pathways or sets of pathways that you might need to get a stem cell to proliferate and differentiate along a specific lineage and fill a hole in bone or grow a new pancreas. Or, in reverse, to shut down a cancer cell mutant in perhaps 5, 6, or 25 separate events and pathways. So we made a model, and the model was, we took these microRNAs, these hematopoietic expressed microRNAs, and we looked at their targets. And on an informatic basis, we said, because a microRNA, say 221, inhibited various targets that were known to be evolved, or at least associated with erythroid development, but not with myeloid development or earlier process in development, we would hypothesize that what this microRNA, or these two microRNAs, were doing was controlling erythroid development. Similarly, a microRNA like 155 that regulated both myeloid and erythroid and early molecules by the thermodynamic predictions, some verified by our luciferase assays, was acting way back here to inhibit both myelopoiesis and erythropoiesis. Well, we could test that. And so we could say here, first in a model system, that we would take the K562 cell line, which when you add hemin to exponentially growing cultures of undifferentiated K562, this is a human leukemia cell line derived from a patient with chronic myelogenous leukemia in blast crisis of acute myeloid leukemia. The blasts have some erythroid features, but they have some myeloid features also. But if you add hemin, these cells actually make hemoglobin, which you can assay with monoclonal antibodies or other techniques, or just do a quick benzidine stain of a slide for hemoglobin. And you see all the brown here on this slide, and if you actually held this slide up, it was brown in the cytospon area. That's the control. If you took and first lentivirally transduced using the two-color molecules that lentiviral vectors that I described earlier, you could overexpress miR-155, and you blocked the expression of hemoglobin and therefore probably the erythroid differentiation of this model erythroid cell line. So what about primary cells? So we did the same experiment in primary cells with the same vector, and overexpression of miR-155 drove down the myeloid and the erythroid colonies, and you can see the few colonies that we got were small and dysmorphic. So it looked like one microRNA verified our predictions, and we could write a paper. What's happened since? And miR-16, we predicted, would, in this system, inhibit erythropoiesis, but not myelopoiesis. And Richie Hildreth, now a PhD student at George Washington, while he was a tech in my lab, undoing all the fun he had in college and getting into a graduate program, he basically did that by showing that miR-16 selectively inhibited erythropoiesis, but hardly, if at all, inhibited myelopoiesis. So the model was verified again in this prediction. We've gone on, and one of the targets of miR-16 is well known to be BCL2, and that's important and subject to deletion in chronic lymphocytic leukemia cells. Was that the target in erythroid cells? Well, actually, there are many other predicted targets, about 100 other predicted targets expressed in hematopoietic cells. And we've shown that 1433 and AKT are also miR-16 targets, and a list of other molecules involved in apoptosis, and also a list of molecules important in differentiation. So we've still got to go through and figure out how miR-16 actually is working in the cell, and various knockout approaches are in progress to do that. Here again is our map of hematopoiesis, which we're busy figuring out how do these microRNAs act, if we're right, as circuit breakers which control the development of all these lineages, and how might we act upon this system, therefore, to control it by following nature. The next thing we looked for was there something interesting in cancer here? And as I alluded to, miR-16 had already been shown to be part of an important process mechanism in chronic lymphocytic leukemia. Were there any tumor suppressor genes or candidate tumor suppressor microRNAs that we saw among the hematopoietic expressed microRNAs, the hemirs? And we just looked at the expression results of our hemirs, and we came up with two, miR-10A and 27A, both indicating that they're both part of families, which will make some of the studies difficult. And we found that while they were quite well expressed in primary CD34 positive cells, they were below background levels of expression. I think actually in one cell line this creeps to just above background, and we've got to investigate that. But in all of the cell lines we looked at, and we've looked at a much bigger panel of this, leukemia cell lines and primary samples, they were not expressed. So are these tumor suppressor genes? by the way, they're also missing in many other types of cancers, but we haven't looked at that systematically yet. Well, where are they in the genome? They're located at potential fragile sites and sites of deletion, so there's a potential association there. And Bob was happy because when he took the informatic tool, the transcriptome interaction database, which he made, he found lots of targets for potential targets for miR-10A and 27A. Among those targets, we've selected a bunch that are involved potentially in apoptosis. And we're trying to go after the hypothesis that these regulate an important apoptotic system. So we took miR-10A, and Kara Scheibner has done a lot of these experiments with Bob. And we first replaced them in the leukemia cells that are missing their expression and found nothing happened. But when we induced apoptosis with a drug in these leukemia cells, either staurosporine or camptothecin, we found that miR-10A transfected cells had exaggerated apoptosis, so that once apoptosis was gotten going, miR-10 contributed to the apoptotic mechanism. Similarly, for miR-27 in another cell line contributed to the apoptosis. And here's a really neat experiment where suboptimal amounts of miR-10 and miR-27 synergized in contributing to staurosporine induced apoptosis in a couple of different cell lines. And so missing both of these in a leukemia cell might be two of the hits that leukemia has. We're a long way from proving that. We're in the process of looking at targets and how this might work, as well as beginning to look at the regulation of miR-10A and 27A. But Kara had another problem in that she wanted to see what would happen if you knock these molecules out of primary hematopoietic stem progenitor cells. So she had to make some anti-miRs, and these are essentially antisense that are made basically non-degradable, but also to have a higher affinity for the complementary molecule. But there was no consensus in the literature as to how you make these, and specifically which bases you should lock to get the higher affinity and lower degradation. And what I'm going to show you is Kara found that if you lock, for 3 of the microRNAs that we've now looked at least, if you lock the 8 bases in the center of the approximately 22 base microRNA sequence, you get good and specific inhibition. And I think I probably don't have time to take you through this data, but she looked at various strategies for which bases to lock, and the 8 in the center strategy has been the winner in the 3 that we've looked at before. So now we can do loss of function experiments, and we can do them in human cells as well as mouse cells. And I'd point out that it's not so easy to make a microRNA knockout necessarily. For some it's easy because they're encoded at only one site in the genome, but many others are encoded exactly, with exactly the same sequence at multiple sites in the genome, and many others still have family members that are different by a base or 2. So we'll get at the problem we had with KF4, KF1, and KF3. We're also moving on to study mouse hematopoiesis because there are a number of experiments we can do there that we can't do in the human system, and because we think that the Venn diagram of the really important microRNAs and targets are going to be the ones that work in both systems. And so Ian Kaplan and Diane Heiser are pictured here, and they focused first on the myeloid axis and purified each of these populations of stem progenitor cells in the mouse, and have done purified huge numbers of them. We call them the bone crunchers because of how many mice they have to kill to do these experiments. and they have looked at expression of RNAs and messenger RNAs and microRNAs. I want to take you in a slightly different direction for the last couple minutes, and this direction is stimulated by the fact that this is a Cancer Institute-sponsored meeting, and that we think that these microRNAs might provide the basis for therapeutic molecules eventually, although targeting them to tissues may be a formidable challenge. Ihor mentioned the cardinal properties of stem cells, and I want to mention cancer cells, stem cells, as no doubt Bob Weinberg will speak about later. In my thinking, cancer stem cells are cells which have the key cardinal properties of hematopoietic and other stem cells in that they can self-renew and they can differentiate. And critically, a hematopoietic stem cell has the potential to generate, as Ihor said, huge quantities of blood cells already. So transforming this into a leukemia, teleologically, might be not all that difficult. So certainly, I think most of us will agree that that cancers arise from stem progenitor cell populations, and we're getting to want to buy the concept that in every cancer, a cancer stem cell sustains the cancer. So if we're going to look at treatment of leukemias and think about treatment of leukemias with microRNAs and other selective molecules, we've got to find out how to assay and eventually make sure that we have successfully targeted the leukemia stem cell in the example that we're interested in. So we had assays already for acute myeloid leukemia stem cells, and it's well known, and we'd use these assays, that the frequency of the model cancer stem cell in acute myeloid leukemia is about 1 in a million, and this is defined by John Dick and colleagues by taking a leukemia and titering its injection into a highly immunodeficient NOD-SCID mouse. But then 2 papers recently in Science that Sebastian Morso, a graduate student lab, noticed have shown that in 2 leukemia models of acute leukemias that actually you could get leukemias when you transplanted as few as 10 or 100 or 2,000 model leukemia stem cells, arguing that one of the key associated properties of a stem cell, namely that it's rare, wasn't true, at least in these models. So what would happen in a different disease, acute lymphoblastic leukemia, that we also wanted to study? So we said, okay, let's study this in a slightly different mouse. There's a new mouse now that has been enhanced from the NOD-SCID in that it has the IL-2 gamma chain knocked out, and this has multiple advantages. It's more immunodeficient, no NK cells. In addition, they don't develop their endogenous mouse T-cell lymphomas that the NOD-SCID strain is susceptible to, and that complicates your analysis. You have to use more mice, and it can confuse you. So we'll use these mice. They have the additional advantage that because they're more immunodeficient, presumably, it takes 10-fold fewer normal hematopoietic cells to generate normal hematopoiesis. Might it take fewer leukemia stem cells to generate leukemias in this system? Might it be a more sensitive assay for us in acute lymphoblastic leukemia specifically, but then we'll have to go back and look at acute myeloid leukemia. So the model is you take these mice, you wait till they're big enough at 8 or 10 weeks of age to tolerate sublethal irradiation, and you can inject their tail veins with leukemia cells and titer in some primary human leukemia cells that have been cryopreserved from patients, either, I'll tell you about, at relapse or at an initial diagnosis. So the first patient we looked at happened to be a patient in relapse who had the Philadelphia chromosome, the BCR-ABL mutation, among other mutations, no doubt. And when we injected these cells in the tail veins of these irradiated immunodeficient mice, in about 5 weeks, the mice looked really sick, anemic, losing their fur, hunchback, etc. And when we looked at their blood, or their bone marrow, or their spleens, first of all, their spleens were massive. These animals have no endogenous T or B cells, so there are just a few monocytes and granulocytes and red cells in their spleen. They're about 1/10 or lower than the normal spleen size. These spleens were huge and often occupied most of the abdomens of the mice. So this was an easy marker for acute lymphoblastic leukemia. We found in several other cases that they got big spleens. The spleen, bone marrow, or blood was composed, essentially replaced. Here it's about 85% in this mouse based on when we killed it, with CD22 positive, CD19 positive. They're B cell markers that were on the initial case. And the case breeds true in that it looks the same by markers and essentially the same by microRNA after expansion, as it were, in these mice. So we now had this mouse, and we titered its dose in from the cell dose that we transplanted from 1 million cells down to 1 cell. And I was startled to find we've repeated this experiment twice with cells directly from the patient and with mouse passage cells. And I was startled that I had predicted that at about 1,000 or 10,000 cells, we would get no engraftment because of the prior results with a frequency of 1 in 1 million cells in AML. But we get 3 of 10 mice got leukemias. You had to wait longer, but got leukemias with just 10 cells per mouse. Now remember, in a patient sample, this is a sample that is taken from the patient, sits around a while, gets preserved, gets thawed. So, and 3 of 10, you leave a few cells in the syringe and needle. So 3 of 10 is a pretty good frequency. And interestingly, when we compared the two models, might this difference that we're seeing be due to the better model that we're using? And to some extent, it is, in that for each dose of cells that we titered into the mice, it takes longer to get leukemias in the non-skid mice than the nog-skid mice, and we do lose some of these mice over the time interval. So you can imagine that if you killed all your mice back here and assessed them, you would say that this case had a leukemia stem cell, but this case, when assayed in this mouse, it had a leukemia stem cell, but when assayed in this mouse, it did not. So on the other hand, if you do the assay patiently, it looks like you'll get the same answer in both mice. So that wasn't the answer. Is it just this one case? Surely there must be cases, and this was from a relapse patient and had BCR-ABL. Maybe it's got a lot of mutations. It engrafts quickly, and so it's so in this case, essentially every cell is a cancer stem cell. So we've taken a couple more cases, primary cases from from relapse, and was it just relapse cases? So let's take some cases that haven't been chemotherapy treated and are presented at initial diagnosis, and recall that these are childhood ALL samples. So these are from patients who are 80 or 85% cured of the leukemias. So although we don't know for these specific patients, we have the permission to find out, but statistically very likely that 2 or 3 of these patients were indeed cured patients. The titering is ongoing. I can't show you the results of all these, but it looks like that at least in a couple of the relapse patients, you can at least get down to 1,000 cells and get leukemias. And the kinetics, there's no suggestion that it won't go down further. So what we're wondering from this model that we were initially just setting up to test microRNAs and other drugs, whether indeed these patients have in ALL, there's a fundamental difference from AML, and indeed if in at least some of these cases in this disease, either there are no stem cells or every cell is a cancer stem cell. So I hope I've given you some things to think about today. I wanted to show you this example of the hype cycle. This is developed by Gartner, and if you go onto the internet, you'll see this, and this is true of computers and cell phones, but I think it's also true of stem cells, in that you get a new technology, like mouse and then human embryonic stem cells, or like iPS cells. You get really, really excited. This is going to do everything for you. This is going to cure every disease. Everybody should switch over and work on it. And then you say, well, you're going to get cancers, you know, you're going to have problems getting the cells. You have techniques that you could only do for a female patient, you know, and all sorts of troubles with the cells. They don't grow very well. They become chromosomally abnormal, get disillusioned. They go so slow that nobody can get his thesis in a reasonable amount of time. And then you turn these around. Maybe now you're starting to work with iPS cells. You're starting to understand a little more about adult stem cells. You're understanding, as Ihor said, the basic processes, not just the rush to transplant the cells. And you're in this slope of enlightenment where I hope that I've helped to convince you that we are now in stem cell research. And so I just want to close by thanking the people that have gone on this fishing expedition with us over the past couple of years. These are Chesapeake rockfish, and these guys are so kind that they even let me reel in a fish or two every once in a while. And here they are in the lab. Thank you very much.
We certainly have some time for questions.
There's our hand over there.
Yeah. There's a microphone coming.
Thank you for a great talk. I'm particularly interested in the microRNAs. And one of the problems is that Croce has reported, and actually so have we, that MER16 is elevated into B-cell leukemia. But yet your MER16 studies are showing that they affect erythroid lineage. Likewise, MER155 is elevated in B-cell leukemias, but it also affects the myeloid erythroid. Can you comment on that?
Well, a quick answer to this, I think, it's a good question. The Croce experiments that I'm thinking of are MER16 and B-cell specific expression. So that it hasn't been assessed in the hematopoietic system, or at least until our beginnings at this. A quick answer to your question is first that there are predicted to be, at least at the moment, only about 500 or 1,000 microRNAs that are expressed, that we found to be expressed at significant levels and that are important. There may be many more that are just at low levels or expressed in some tissues. But maybe, since there are so few and since they have such a big job to do, maybe nature has used them at multiple places, perhaps with different targets. So the target of miR-155, the famous miR-155, miR-155 is one of these microRNAs that there's just a single copy of in the genome, so it's been knocked out by Rajewski and another group. And when they knocked it out, they got no lymphoid cells, and based on their target analysis, they suggested because this was an effect of miR-155 and various cytokine levels necessary for BT cell development. That's far from shown, and because they did that in a B cell I'll take the back. So is nature using these at a lot of different places and with different targets? So whereas in the B cell, it may be important to regulate this, and this cytokine, and that's how miR-155 or miR-16 works, in addition to apoptosis of miR-16, that in early hematopoiesis, it might work quite differently, particularly if those targets aren't expressed. Now that's assuming we're right, and that the, as it were, translational targeting is the important thing. but then you come in with the transcriptional targeting, and maybe the mir is sometimes the reason that you don't have the message detected on your array. So it's very complex, determined not just by the microRNA, but by the levels of its targets, perhaps at least for the so-called translational regulation. Of course, I haven't answered your question. It's a good question, and maybe your experiments or ours will answer them in the future. Sir.
I think there's a microphone coming. Sorry, Jim.
I enjoyed your talk as well. I was wondering, to answer that question, have you looked at the 5' genomic sequences of your mirrors of interest and see if there's any clues that the chromatin environment might give better insight into the lineage-specific expression?
Well, I talked to a young man last night who is doing just that. The reason we and others haven't solved that yet is the depressing information that the promoters of mirs are often 30 kb upstream. So it's formidable informatics and wet lab work to identify the promoters of mirs. But clearly, that has to be done. I've shown you more on the target end, which is imperfect and confusing at the moment. The control level at the promoter end, it looks like they're classical transcription factors and classical mechanisms, but they work at great distance, unfortunately, so it's going to take us a while to figure that out. There was a question over here.
Here. I think you mentioned that the best way to knock down microRNA is to target the 8 bases in the middle. And I think it's generally, there's a general belief that the most important part of the microRNA in finding its target is the 5' end. Yeah. So can you comment on that? So
as you know, but just for the audience, microRNAs bind to a seed sequence of about 8 bases. There's perfect recognition of this seed sequence, but then generally there's a loop of non-recognition, and then in the rest of the 22 or so bases, -8 and minus the loop, there'll be some homology, 2 or 3, 4 or 5, it depends, and you can use computer algorithms to figure out thermodynamically whether that should be a strong binding target or not. So I think the questioner, who I can't see, but he is asking, it's a man, he is asking, why don't you just inhibit the seed sequence and block there? And when we tried that, it's unspecific. And remember, the seed is just 8 bases. So there are a lot of copies of those 8 elsewhere in the genome, and specific copies that are in 3' UTR recognition sequences. So basically, that did not work as well. Nor did it work to make just the 8 center antimirs that we talk about. Just making those 8 didn't work. We had to tail them and have the full 22 or so.
2 questions. Do you think the stemness of your ALL patients reflects CD34 levels? And secondly, is this going to be a prognostic kind of a test in terms of numbers of stem cells?
Well, we have one case where we've shown this 3 and 10. So I guess to answer you, we've got to wait a couple months for our, at least to have 6 or 7 cases. I didn't get to tell you, but you may have noticed that although the fastest case generated leukemia in these mice in 5 weeks, the slowest case took 7 months. So you can imagine, that's with 1 million cells. You can imagine how long it's going to take us for some of these. And the the samples from patients at initial diagnosis, untreated, tend to be the long ones, and they're the most interesting ones to us. So whether then there are subsets if perhaps the initial diagnosis cases or the cases with known prognostic markers like CD34 or something else, or the known molecular abnormalities, maybe the cases with BCR-ABL will generate leukemias faster and at lower doses than the others. Our working hypothesis and hope in the lab now is that every case will generate leukemia at a very low dose, but we only have proof of that for one case, and so right now you can bet on either end, and I'll take your bets.
So do you think it's, oh, sorry. So is this peculiar to the particular disease you study, which is basically a stem cell leukemia, and at least it's assumed to be close to stem cell leukemia, or you think it's a problem with the assay? And if you go back to the older studies, that actually all tumors would do this.
Yeah. Well, just to clarify for the audience, because when you have the BCR abnormality, of course, that's paradigmatic for chronic myeloid leukemia, which we all accept as a stem cell disease. On the other hand, when you have the BCR-ABL fusion gene in acute lymphoblastic leukemia, that's, it's not like CML at all. It's a different disease. It happens to have a very similar molecular abnormality. So still, is this leukemia, in this one case, a stem cell leukemia? And the others that are the more curable, TEL-AML1 or the other mutations that go along with ALL, will they not engraft at a low level? You saw every case we've tested engrafts down to 1,000 cells. When we were planning the experiments, we didn't think there was any point in using lower doses than that. So now we've got to patiently wait for the mice to yield tumors or not at the lower doses. It's a good question.
Do you take seriously the fact that when you did the dilution curve, you had a linear function? The reason I ask that is, there's a hypothesis that hasn't been put on the table, and that is that there are stem cells and there are non-stem cells in the population, but when you dilute them back and incubate them in a mouse in this kind of environment, you can move in either direction. You can move backwards in the direction of development and create a stem cell by virtue of the experiment. Right? Now, you wouldn't see that as a linear dilution, and the reason I say that is because obviously if you put in a lot, it wouldn't need to differentiate, and you would see a function that's not linear. So that's why I ask, do you take seriously the linearity of the dilution curve?
Well, it's linear, it's log linear actually, but it's log linear not just in that one case, but if you plotted the others for which we have partial data, it's also log linear. So you can look, and if your mice get leukemia at 5 weeks in this first case, case 1, at 1 million cells, at 100,000 they'll have leukemias the next week. And by the way, if you test their blood, they have leukemia way before that, of course. But we use this assay. And it does look like it's characteristic. So these cells are behaving like, at least in the mouse, like ping pong balls in a math analysis, not like something that leads 2 or 3 or 4 of them or some interaction to go. It's fascinating to me as a pediatric oncologist that you can get a case of a fully evolved leukemia, and you take 1 million of these cells from a patient who's very sick from this disease, and you put them into a mouse, and it's 7 months till the mouse has leukemia. Now, we need to label these cells and watch them in imaging as well as sampling the tissue and see what's happening. Is the growth like that, linear or log linear? Or is the growth like this? Or is the growth only in one site for a while? Are there quiescent periods? This will be a fun model to look at.
Thank you very much. We're going to take a